course on medical biomaterials, we will talk about biological responses. In the previous class also I talked about biological responses. We will spend some more time on the biological responses, because as soon as a foreign body, whether it is a metal or whether it is a polymer or a ceramic, placed inside the system, short duration, long duration, immediately the defense mechanism starts acting, and this defense mechanism may involve antibodies, coagulation factors, inflammatory factors, the formation of encapsulation of the biomaterial. So many things can start happening actually. Some of them are felt only when the material is placed for a very long period, and some of them are felt immediately as soon as the material is placed within a short duration. So, we are going to spend some more time on this, because this information is very important, so that to be very sure that the material does not cause cytotoxicity or any other problems to the host. And before going into a human, these materials are always tested in animal, and they perform all these tests to identify all these issues. Okay, so what happens? So, what are the various responses that happen inside the coagulation? So, many factors related to the blood coagulating factors come into picture here. Okay. Then the complement, there is a set of proteins which help in trying to attack the material. This is the host defense system. Okay. So, there are some complement proteins which tries to identify this foreign object, and there are some complement proteins which tries to attack them. So, all those things will happen. Then inflammation starts happening. The inflammation mostly around the site of implant. Inflammation is good because it helps the host systems to adjust as well as attack the foreign body. And then you also end up having capsule formation. So, the formation of giant cells, multinucleated cells, all those things start happening actually. So, these are the various responses that happen. As I said, some of them start happening very fast within a few hours, and some of them start happening within a few days, and so on. So, ultimately the goal is to achieve biocompatibility of the material and try to address the cytotoxicity or genotoxicity of the material. So, the material has to be both biocompatible and it should not cause adverse reaction. So, that means it should not be cytotoxic. Okay. So, in order to understand this, I thought we need to spend some time on the various biological responses of the biomaterial to the system. Okay. So, we will spend some of them, and I did introduce some of these topics in the previous lecture as well. Okay. So, there is sequence of events that happen as soon as the biomaterial is implanted into the system. So, the place where it is implanted is considered as a site of injury. So, there could be a blood material interaction. If it is a cardiovascular stent or if it is a diaphragm valve, there is going to be a blood material interaction. Okay. Whereas if it is an urethral region, of course this is not going to be happening. Then there is a provisional matrix formation. Okay. So, if there is a blood material interaction, there could be coagulation, things start happening. Okay. There could be other things like complement activation could be happening. Then you have the acute inflammation, that is at the site, then it leads to chronic inflammation. Okay. Then formation of granulation tissue, foreign body interaction, and finally the material could be completely encapsulated, that is fibrous capsule development could happening. So, there could be inflammatory activation, and there could be immunological activation. So, you could have immunological things happening immediately, then inflammatory actions happening, and then again immunological activities happening later actually. And that is how the system, the human system or the host system starts responding to this biomaterial. Okay. So, we need to understand this, and whether this leads to complications to this host needs to be also recorded. So, these are related to the tissues. So, inflammatory wound healing responses. So, it is like an external injury a wound. So, there could be several sets of responses which are called wound healing responses, and there is a foreign body interactions, okay, because you have placed a material which is a foreign body. And then finally fibrous encapsulation. So, when a biomaterial is placed, after a few months or few years, it will be completely encapsulated by the tissues, and it may be impossible to remove the material from the host at all, because it is completely encapsulated. Okay. So, if somebody wants to remove, especially if there are certain joint failures, okay, and then they want to replace it and place a new joint, the surgeons find it very difficult, because the material is completely encapsulated. Even if you take cardiovascular stents, it may get encapsulated by tissues. So, it may be very difficult to remove that old stent and try to place another new stent. That is why there is now a lot of interest in looking at biodegradable stents, so that the stent will completely go away. So, if the cardiac surgeon wants to place another stent in the vicinity, he or she will not have any problem. Okay. Now, let us look at each one of them a little bit more in detail. So, there could be 4 types of tissue responses here, because there could be different types of material that is coming into the human system. Toxicity of the material is very toxic. Okay. That means the material itself may be toxic, or there could be some leachants that could be toxic. There could be some acids getting leached, lactic acids, or acrylic acids, or some other maybe silver nanoparticles. All this could be very toxic because of the local concentration gradients. So, there could be death, cell death. Okay. So, this we can even look at it in vitro. For example, I talked a couple of classes back an assay called MTT assay, where we can look at the viability of cells. That means how many percentage of cells that are viable with respect to control. So, we can incubate the biomaterial with the cells like L6 or 3T3, which are mostly muscle related cells, and then after 24 hours we can count and see what is the percentage of cells that are live. So, that is called an MTT, where we are using a reagent MTT, or we can look at cytotoxicity and so on. Actually, there are so many ways to determine whether the cells are dying, whether the cells are dying naturally. When the cells die naturally, we call it apoptosis. When the cells die because of a foreign material, then it is called necrosis. So, we can monitor whether the death is apoptosis or necrosis. If the cells die because of necrosis, the cell membrane is damaged. So, the material inside, the enzymes inside are leaked out. So, we can find out what is the enzyme that is coming out, for example, LDH type of enzymes, okay, which gets leaked out when there is necrosis. So, this can be easily studied through in vitro. We can look at different cell which are related to the human system, okay, and then find out the toxicity of the material. If you have non-toxic nearly inert materials, okay, for example, PTFE, originally if you look at biomaterial history, the second generation, it was felt can we have inert material as biomaterial. PTFE, PVC, they are extremely inert. Okay. So, what happens? Formation of fibrous tissues with variable thickness. So, you are going to have the entire material encapsulated by fibrous tissues. This is called fibrous capsules around, this could be soft and hard tissues. That was in the second generation biomaterials. It was always felt and the biomaterial should be completely inert. Okay. PTFE is supposed to be very good, titanium is supposed to be very good. So, small diameter vascular grafts completely inert. Okay. So, such materials are non-toxic. So, there is formation of fibrous tissues, as you can see here. It could be a thin fibrous soft tissues or hard tissues or thick, and it completely encapsulates your material. Now, non-toxic bioactive material, that means the material is active, it is not inert. So, there could be formation of interfacial bonds between the tissues and the biomaterial. Okay. So, it is not completely engulfing or encapsulating your biomaterial, but it is forming interfacial bonds. It is very difficult later on to remove if there is a fibrous encapsulation or even when it is form an interfacial bonds, because the material is completely become part of the host system. Fourth type, non-toxic dissolving. For example, it could be a biodegrading material or biosorbing material. So, what happens after some time, maybe a year, 2 years, the material is completely disappearing, dissolving, like your PLA, okay, or some PLGA and so on. So, what happens, that area is getting replaced by the surrounding tissue. So, the tissues start growing as if the material is not there, material has completely gone. Ideally, it could be used in tissue engineering. So, I am growing tissues with some polymers which are bioresorbable, then you are placing it inside and the tissue grows, material disappears. Maybe one day we will have the cardiovascular stents which is completely bioresorbed. So, the cardiovascular stent may completely disappear, unlike the current stents which are made of nickel titanium. So, 4 types of situations: toxic biomaterial or leachants from the material that will lead to cell death, non-toxic nearly inert, so there could be fibrous encapsulation, or non-toxic bioactive, there could be formation of interfacial bond, non-toxic dissolving material, so it is replacement by surrounding material. But here we may say non-toxic dissolving, there could be some toxic dissolving material also. For example, polylactic acid, it is dissolving or it is bioresorbing, but the lactic acid that is produced is little bit toxic. So, there could be some cells dying in the vicinity, there could be little bit local toxicity because of the local acidity. So, there could be some variations here, toxic dissolving material. Okay. So, we have different situations and the tissue responds in different ways. That is the beauty of our defense system, which looks at coagulation, which looks at complement activation, mainly to attack the foreign material, leads to inflammation, the chronic and acute inflammation, then it leads to fibrous formation, encapsulation, giant cell formation, all those things happen, and you need to know that such things can happen. That is very important for us to understand. Let us look at each one of them little bit in detail. So, biocompatibility, as I mentioned, the ability to provide an appropriate host biological response in a specific given application in the body. So, specific application. So, we have different applications like we have been talking about. It could be short duration, long duration, permanent stay, it could be in blood contacting, non-blood contacting, urinary region, it could be a inert biomaterial, it could be an active biomaterial, it could be a bioresorbable material, it could be a metallic which does not so, given application, so it should have appropriate host response. Okay. The material should not create any adverse biological reaction. We do not want the material to lead to systemic toxicity or acute toxicity, okay, genotoxicity, or metal leaching, which could be problems to the host, okay, creating certain side effects. So, it could cause allergy. Metal leachants can be allergic to the patient, or lactic acid leaching out could be allergic, acrylic acid leaching out maybe from dental implants could be allergic. Cytotoxicity, cell culture cytotoxicity, that means the toxicity of the material to the cells, especially the addition of the cells to the surface, proliferation, activation and death. So, you want the cells to adhere, proliferate, get activated, and finally it should go through the apoptosis, not through necrosis. So, the biomaterial should be compatible as well as not create cytotoxicity. Okay. So, look at the biological responses. Let us go a little bit in more detail. So, adsorption of blood proteins is happening here. If it is a blood contacting device, then we have activation of the coagulation cascade. This is a fantastic cascade. So, there are many coagulation factors, one after another keep on happening, and finally you are going to have the coagulation. Then you are activating the complement. These are set of proteins which get activated so that they are the host defense mechanism. Activation of the platelets. Okay. So, all these things happen actually. Then we are having activation of polymorphonuclear leukocytes, monocytes, and then macrophages. Okay. These are macrophages are certain cells which tries to form the encapsulating of the biomaterial. Then danger signals, that is called alarmins released from the damaged tissue. Okay. That is the next step. Immune cells show enhanced function via pattern recognition receptor. So, they are able to identify that the material has certain pattern. So, there are some factors getting released. Okay. All these are called immune response. Okay. Activation of coagulation, activation of complement, activation of platelet, then it is activating the polymorphonuclear leukocytes, okay, macrophages which is involved in the engulfing or the encapsulation of the biomaterial, then signals are produced, and then all these immune cells show enhanced activity. Then comes the inflammatory things. You are going to have acute inflammatory response, activation of monocytes and macrophages, okay, activation of monocytes and macrophages, then chronic inflammation happens after the acute, and then you are getting foreign body giant cells, fibroblast activation, and finally macrophage derived cytokines and pattern recognition receptors, engagement activate dendrite cells, all these start accumulating on the biomaterial. So, again immune response takes place. So, we have the immune response to start with, okay, then we have the inflammation which is acute and chronic happening, and then we have again the immune response, foreign body, giant cells forming, fibroblast activation, macrophage derived cytokines, all these things start happening, okay, as the material is placed inside the system. This is the time of events, could be several hours here. Okay, so let us look a little bit in more detail each one of these items. So, we are going to have two things happening when the material is in contact with the blood, the complement and the coagulation. So, what is this complement? This is a system of plasma proteins that is activated by the presence of pathogens, antibody mediated bactericidal activity. Okay. So, these are formed when there are any pathogens, these are formed when there are any foreign bodies, okay. They get activated, and there are 30 distinct plasma and membrane bound proteins. These proteins are found in plasma, these proteins are found in membrane. So, what is their job? They try to identify these pathogens, they try to identify these bacteria, they try to identify this foreign body, so that they can be attacked by the host immune system. Okay. So, there are 30 proteins here, that is what is this complement means. Okay. Then next one is the coagulation. This is a series of calcium dependent proenzyme to serine protease conversions localized on the surface of the activated cells. This is in vivo it happened. What happens? Finally, there is a formation of thrombin. Thrombin is an enzyme which converts fibrinogen to fibrin. Fibrin is a insoluble blood clot. So, when there is a wound, blood comes out after some time, it starts clotting, right, that is called fibrin. How does it form? It forms from fibrinogen into fibrin. This conversion happens because of this enzyme called thrombin. It is an enzyme, thrombin, which converts fibrinogen into fibrin, and this is what is blood clot. So, if this thrombin is not formed, blood will start oozing out, it will not clot. Ultimately, you need to form this fibrin. Okay. For example, you can see this blood clot on a polymer or a biomaterial surface. So, both are very important. So, as soon as the material comes in contact with blood, complement system gets activated. That means about 30 proteins get activated, which are involved in identifying pathogens or bacteria, which tells the defense system of the host that there are some unwanted foreign body present. Okay, and this is the complement. Then the blood starts coagulating. How does it happen? There are a series of actions taking place, and finally we end up in the formation of an enzyme called a thrombin, which converts the fibrinogen present in the blood to fibrin. Fibrin is insoluble, so it is the blood clot. So, two very important actions take place. Okay. So, you need to understand as soon as the material comes in contact with the blood. Now, this complement, there are 30 proteins involved in the complement. Each protein has different type of activity. Okay. So, we will look at that later. So, coagulation. So, I said thrombin is here, which converts fibrinogen to fibrin. This fibrin is an insoluble. So, this is called the blood clot. Okay. This is the blood clot. So, thrombin is a protein which is doing the job of converting fibrinogen to fibrin. But look at this, before that there are so many factors which lead to the formation of thrombin, which is formed from prothrombin, which is formed with the help of factor XA or 10A, 10, 9, 9A and so on. Okay. One is called the intrinsic pathway, other is called the extrinsic pathway. So, two pathways through which you have this particular factor 5A happening, which catalyzes the conversion of prothrombin to thrombin, and thrombin is the main enzyme which converts your fibrinogen to fibrin, and which leads to the formation of stable fibrin, which is in our colloquial term call it blood clot. So, these are series of things happening, and of course, formation of blood clot is very important for the survival of the human being. If the blood does not clot, there could be continuous oozing of blood, and the person can lose blood and end up having a shock because of shortage of blood. Okay. So, especially if somebody has cardiovascular problem, they are given drugs like warfarin or even aspirin, which helps in the thinning of blood. Okay. So, those people who take warfarin or even aspirin have the problem of the side effects. That means, if they have injuries, there could be lot of blood loss. So, they have to be very careful. They can even have internal bleeding because these factors are affected, or these factors are retarded or inhibited by those drugs, especially those who are taking drugs related to cardiovascular for blood thinning. Okay. So, coagulation is very important for the survival of human being, but then when you keep this type of biomaterial, they also lead to some of this clot formation. So, you do not want your biomaterial to initiate this cascade of events and ending up with formation of thrombin, which catalyzes the conversion of fibrinogen to fibrin. Okay. So, it is fantastic set of things happening inside the human system. Okay. I did talk about complement. Okay. These are the system of plasma proteins. So, they also get activated, which are involved in so many antibody mediated bactericidal activity. So, we will look at that. Complement pathway. There are again 3 pathways here: classical pathway, lectin pathway, alternate pathway. Okay. And as I said, there are almost 30 distinct plasma or membrane bound proteins. So, they all get activated at different sequence of events, and they in turn activate eosinophils, basophils, monocytes, macrophages, so many things they can activate. Once they get activated, they can even kill the cells. So, this cell lysis also takes place. So, so many different types of complements, and they end up killing cells here, inflammatory and cell infiltration. Okay. So, we do not want the complement activation also, which may lead to cell death. Okay. What are these So, main consequences of complement activation, opsonization of pathogens. What is this opsonization? These complements are fantastic proteins. They identify what are the pathogens so that the host defense can go and kill them, is not it? So, they are like your in the war special force which identifies who are the terrorists. Okay. So, that is what it does, opsonization of pathogens. So, some of these complement proteins, our job is to just identify pathogens, that is one. Some of the complements, recruitment of inflammatory and immunocompetent cells. So, they will start collecting inflammatory and immunocompetent cells towards the target size. They all want to go towards the place where the biomaterial is placed. And third is direct killing of pathogens. They directly go and they will go and kill the pathogens. It could be a bacteria, because the biomaterial is always considered as a foreign object. So, for the immune system they are unwanted. Okay. So, three things these complement proteins do. They identify the pathogens or the foreign body to the host defense. So, the host defense once they are identified, the host defense system can go exactly and kill. So, it is like the special force goes and identifies who are the terrorists in a village, and the army comes and exactly targets those people and leaving the innocent people. That is the job of complement. They recruit inflammatory and immunocompetent cells. So, they bring these cells towards closer to the foreign body. They directly kill the pathogens. Okay. So, there could be pathogens killed, so some good cells also get lysed and so on. So, all these are the main activities of these complement. So, once they get activated, they start doing these three different jobs. Okay. So, we do not want that to happen, especially when we place a biomaterial. That is why especially when a patient undergoes a implant, they try to give drugs so that the immune system is slightly suppressed. We do not want the immune system to become active, creating blood clots, complement activation, and then systems becomes inflamed and so on. So, they give a lot of drugs where the immune system can get little suppressed. So, the patients can have the risk of having diseases where because the immune system is compromised here. Okay. So, they can have bacterial infection, they can even have long term HIV type of infection because their immune system is suppressed. Okay. So, these are the job of the complement proteins. So, one needs to keep in mind when the biomaterial is placed that the biomaterial does not activate the complement proteins. At the same time, we do not want the biomaterial to activate the coagulation pathway because there could be a lot of blood clotting taking place, as I have been explaining, formation of stable fibrin which are insoluble around the biomaterial, formation of clots. So, we do not want both these things happening when the biomaterial is placed inside the blood clot acting area. Okay. Okay. So, these are many complement proteins. Okay. These C3, C4, as I said, there are 30 different plasma bound or membrane bound proteins which are called complement. These proteins C3 and C4 identification, that is opsonization of pathogens. C3a, C5a, they are recruitment activation of inflammatory cells. C5b9, lysis of pathogens cytotoxicity, C5b9. So, you are looking at cell lysis. C3, C4 are involved in identifying the pathogens. C3a, C5a are involved in the recruitment of inflammatory cells, sorry, C3a, C3b recruitment of inflammatory cells, clearing immune complexes in apoptotic cells. So, once the cells have died, you need to remove this, that is C1q and so on, okay, C1q, and then augment cellular immune responses, again bring back the cellular response with the help of T and B cells. So, C3, C4. Okay. So, these are the jobs of these complement proteins. Very interesting. Such fantastic things are happening inside the host system. So, I think it is very fascinating. The complement system, it identifies the pathogens, it brings the inflammatory and immune competent cells, it directly kills the pathogen. So, all these three things are done by the complement system. Okay. So, complement activation, okay, how does it happen? So, we can or how can we prevent this complement activation? So, we can have modification of device materials or the blood contacting materials. People have looked at heparin coating. Heparin, by coating this heparin, it will prevent both complement activation as well as it can reduce the coagulation of the blood. Okay. So, this can limit complement activation and subsequent inflammatory response. So, that is modification of the devices, heparin coating. So, there is a lot of interest nowadays on heparin coating. There are some polymeric material which are moderately activating the complement based on cellulose, like cellulose acetate, hemophan, cellulose triacetate, all these biomaterials moderately activate the complement system. There are some biomaterials which very low activating, polymethyl methacrylate, polysulfones, polyacrylonitrile. That is why polymethyl methacrylate is widely used in the oral or dental. Polysulfones are also used in some of the guide wires, guide tubes, okay, because polyacrylonitrile also are very useful because none of them activate the complement system. They are all synthetic material. So, one needs to keep that in mind if one is designing biomaterial where blood contacting biomaterial and we do not want the complement activation. So, one is modifying the surface, and heparin is widely nowadays used which can prevent both the coagulation as well as complement, or we can think of using these type of natural material which moderately activate, or these type of synthetic material which does not activate the complement system. Okay. So, we will continue more on these biological responses in the next class also. Thank you very much for your time.